Hi everyone, in this second video on Advanced Pharmaceutical Dissolution Science and Application series, we will discuss biopharmaceutics studies, how a drug's formulation, physicochemical properties, and route of administration affect its absorption and therapeutic performance. It connects formulation design, drug release, and drug absorption, making it the foundation for understanding dissolution. In simple terms, biopharmaceutics explain how a drug in tablet becomes available for absorption in the body and how formulation choices translate into in vivo performance. It answers three key questions: how much drug dissolves, how fast it dissolves, and how effectively it reaches the bloodstream. That's why dissolution testing is a key biopharmaceutical tool. Now, biopharmaceutical pathway. After you take a tablet or capsule, three key steps occur. First, the dosage form disintegrates into smaller particles. Next, the drug dissolves in gastrointestinal fluid. Finally, the dissolved drug is absorbed into the bloodstream. Dissolution is the critical middle step. It controls how quickly the drug becomes available for absorption. Next, for any oral solid dosage form, tablet or capsule, dissolution is the first essential step. A drug that does not dissolve cannot be absorbed. Poor dissolution is one of the leading cause of formulation failure, batch rejection, and even clinical trial setbacks. Even most potent drug is ineffective if it cannot dissolve. That's why for many poorly soluble drugs, dissolution becomes the rate limiting step. By understanding dissolution, we can better predict absorption and overall bioavailability. This is exactly why dissolution is monitored from early development through post-approval. Further, dissolution testing acts as a window into how a drug behaves in the body. Although it is an in vitro test, it is closely linked to in vivo absorption. Even small changes in API form, particle size, or excipients show up quickly in dissolution results. That's why regulators rely heavily on dissolution testing. In general, better dissolution leads to better bioavailability, especially for BCS class 2 and 4 drugs. When dissolution is slow, absorption becomes slow or incomplete, resulting in lower plasma level and reduced therapeutic effect. This is why dissolution testing is a powerful tool in biopharmaceutics and in predicting in vivo performance. Now we will discuss BCS, the Biopharmaceutical Classification System, or BCS classifies drugs into four categories based on solubility and permeability. BCS helps us understand how critical dissolution is for given drug. For class 1 drug, dissolution is usually rapid and not the rate limiting step. For class 2 drugs which have low solubility, dissolution becomes the biggest bottleneck to absorption. In fact, most modern drug falls into class 2 and class 4, which is why dissolution testing continues to gain importance. In simple terms, class 1 drugs dissolve easily so absorption is fast. Class 2 drugs dissolve poorly, making dissolution the key limiting step. That's how BCS helps us judge the importance of dissolution for each drug. Further, let's quickly connect BCS classes with the role of dissolution. BCS class 1 dissolution is fast, so it's rarely the rate limiting step. BCS class 2 dissolution is the main barrier. Improving dissolution directly improves absorption. BCS class 3 permeability limits absorption, but consistent dissolution is still essential. BCS class 4 Both dissolution and permeability are challenging, making method development more complex. This framework clearly shows how dissolution impacts biopharmaceutic behavior across drug classes. Next is why dissolution testing matters. Dissolution testing ensures that every tablet behaves consistently. It can quickly detect small variation in compression force, granulation, or coating defects. In generic drug development, dissolution profiles are critical for demonstrating similarity to the reference product. In formulation R&D, dissolution testing helps scientists understand how excipient influence drug release, which strategies improve dissolution, such as particle size reduction, surfactants, solid dispersion, or buffer selection. Whether two formulations are likely to be biorelevant. In this way, dissolution guides key decisions long before clinical trials begin. In practice, dissolution test is considered as surrogate for bioavailability. Regulators like FDA and EMA often accept dissolution data as a surrogate for in vivo studies in specific cases, especially for BCS class 1 drugs. This can eliminate unnecessary bioequivalence studies, speed up development, and reduce cost. When designed properly, a dissolution test can reliably predict clinical performance. Next, we will discuss in vitro in vivo correlation (IVIVC). IVIVC describes relationship between in vitro dissolution results and actual drug absorption in humans. A strong IVIVC means that when dissolution improves, bioavailability also improves, and when dissolution slows, absorption decreases. This makes IVIVC especially valuable for controlled release and modified release formulations. Now we will discuss stability and change control. Dissolution testing during stability studies helps detect early signs of polymeric changes, excipient degradation, changes in tablet hardness or coating, and moisture impact. Dissolution testing also plays a key role in supporting post-approval changes, whether in formulation, equipment, or manufacturing site. I have seen real industry cases where a small change in excipient grade caused a major shift in the dissolution profile, putting bioequivalence at risk. For modified release formulations, dissolution testing even more critical because it directly controls the drug delivery rate. As a routine QC test, dissolution ensures that every batch releases the drug consistently, meets the label claim, and performs like the clinical batch. Any deviation in dissolution can signal problems with raw material, process parameters, or stability. Further, USP recommends several dissolution apparatus types. For most immediate release tablets, the paddle method is commonly used. For complex dosage forms such as pellets or poorly soluble APIs, often requires flow-through cell system. Choosing the right dissolution apparatus is not just a lab decision, it's the key part of overall biopharmaceutic strategy. Next, regulators treat dissolution as a critical quality attribute. That's why dissolution method must be discriminatory. They should clearly detect meaningful change in formulation or process. Dissolution plays a role throughout the product life cycle, from regulatory submission and method validation to stability studies, scale-up, and post-approval changes. Now, I would like to summarize this presentation. Dissolution testing is one of the most powerful tools for biopharmaceutics. It ensures consistent product performance, helps predict how a drug behaves in the body, and forms the backbone for bioequivalence success and regulatory acceptance. Simply put, to understand drug performance, you must understand dissolution. Thank you for watching. Please subscribe us for getting update on such industry application oriented videos and stay tuned because in this 62 video series on dissolution science we will cover dissolution method development, validation, troubleshooting and many real industry examples. In the next video we will cover USP apparatus 1 to 7, when to use which. If you have any question, please post them in comment section. I will do my best to respond. See you in next video. Thanks.